It's always an honor to close this conference, and I look forward to this talk more than anything else, and I've heard it. This is probably the 9th time, but I know no one better to do this than Tony Komroff. He has the best oversight and wisdom and knowledge, I think, about this disease of anyone I could think of, and he does a great summary. He is a Harvard professor of medicine and with a special interest, as you all know, in CFS-ME. And Tony, you're on one more time.
Thank you, Dan. So this is a challenge. There have been something like 170 different presentations, oral and poster, and that covers not just a lot of different disciplines, but a lot of different topics within biology. I'm going to try to pull together what I heard into these general areas: diagnostic tests or biomarkers, post-infectious fatigue, infectious agents, the role of stress, links of CFS to other diseases, brain studies, cytokine studies, and a little bit on treatment. A couple of initial apologies or explanations. Obviously, in 45 minutes, I could only summarize a handful of the excellent work that has been presented over the last 3 days, and that is arbitrary. When one of your slides seemed worth highlighting, the conference organizers loaned it to me, and I've sometimes changed it to make it, to highlight a particular point or to make it more visible to the people in the back of the room. So I hope that I will be accurately reporting your data and the inferences from it, and if not, don't hesitate to let me know. I think we're talking about a lot of very specific issues in the biology and psychology of this disease, but behind it all is still, as Fred Friedberg and other people have alluded to over the last few days, there is still in the minds of some people the question, since this syndrome is defined just by a bunch of symptoms, is there any objective biological marker that is different in patients with this illness? And if so, could that marker be related plausibly to the patient's symptoms? In other words, as the word is used by some people, is CFS a real illness? Now, in my mind, the answer to that has been in for at least 15 years, but much of the work presented at this meeting, I think, also answers it pretty loudly. Also, I want to begin by some context, since research in this field is following the track, not surprisingly, of research in all biomedicine. The last decade has witnessed the most explosive growth in potent technologies for understanding biologically based illnesses in our lifetime. Just a decade ago, we knew only a few of the human genes. Now we know all roughly 22,000 of them. A decade ago, linking a gene to a particular disease or syndrome was like finding a needle in a haystack. Today, it is literally exponentially easier and faster and cheaper to link a gene with a disease. And finally, in the old days, we could really only study the action of a few genes in any particular cell. Today, as we've heard repeatedly, there are technologies that allow us to determine whether all of the genes in the cell are in a state of being turned on or off, and relative degrees of both of those. That's an enormous set of technologies to help understand diseases, and a lot of the work presented here talked about that. We're going to start with several papers that dealt with whether there is some diagnostic test or biomarker that distinguishes patients with CFS from various comparison groups, either healthy controls, depressed controls, other fatiguing illnesses. This was Dr. Fletcher's work and Dr. Klimas's work, plasma neuropeptide Y, and the levels were found to be higher, NPY levels higher in the patients than in the controls. Not only that, those levels correlated with lots of different measures of mood and perceived stress and cognition, and did not correlate with a reasonable screening instrument for depression. So it seems to say that NPY may be linked in some way to some of the symptoms that constitute part of this syndrome. Very interesting work presented by Dr. Light, interesting in part because it was one of the few papers that, for me anyway, came totally out of left field. I've never heard anything before, certainly in this field, about these various ion channel receptors that sense muscle pain and fatigue. And he identified several of them, several adrenergic receptors in immune molecules that also are responsive to muscular pain and fatigue, and then studied them in patients who were healthy, and why isn't that showing? Yeah, okay. Patients who were healthy after an exercise challenge. So this is over a 48-hour period. All of these different markers, biomarkers, are arrayed over here. Several of them, but not all of them, bumped a little bit following the exercise test and over the next several days of recovery. In patients with CFS, conversely, here with the normals, and here on exactly the same scale, there's no difference in the scale, the results in patients with CFS. And as you can see, a much more dynamic response of lots of these different molecules to exercise. So it certainly seems to be saying that for reasons we don't yet understand, it begs the central question of why this phenomenon, but it seems to be saying that exercised muscle responds very differently in these patients. It's tempting to correlate that with the phenomenon of post-exertional malaise that we are all familiar with. That correlation has not been established by this work. Oh, I see what he was doing. Okay. These levels, but the connection was alluded to by these studies of correlation between various subjective measures of physical and mental fatigue with several of the molecules, likewise with pain. So suggesting that there may be a connection between this phenomenon of exaggerated expression of these molecules and the symptoms that are part of post-exertional malaise. Using 9 different genes, or 4 genes and then 9 genes, Dr. Light's group was able to separate pretty nicely healthy controls from CFS patients. So here we have a candidate for a discriminator, not a perfect discriminator, clearly, but one that comes closer than a lot of previously reported tests. Broderick and Fletcher, Vernon, and Klimas studied Gulf War illness patients who meet a somewhat different case definition, but as one of the presenters today said, a definition that has considerable overlap with chronic fatigue syndrome, and measured the expression of multiple cytokines and neuroendocrine markers simultaneously at rest, at peak exercise, and after peak exercise. And then used very sophisticated computational techniques to really extract all of the information that you could out of this huge data set, to see whether you could develop a model off these data that predicted caseness, that distinguished patients from controls.